All right, hello everyone. So, in this video, we are going to be looking at infectious diseases. It's one of the series of classes that we're having on, infectious diseases. Today's focus is going to be viruses, and let me show you what some of your responsibilities will be as we move through this. So, you will find where I posted this video a series of, well, actually just one file other than the file for the video with this worksheet to work through. And as it comes up, I would like you to pause the video and use it to finish this worksheet. There will be a due date assigned, and this due date will have you post it back to me. Either you can print it out and draw on it, or possibly, and try this out, possibly digitally modify the file and send it back to me. Soon after that, I will post to you a key, the expected outcome of your work. You will get credit for the work that you did without penalty for messing things up, as long as the effort was there. The key is there so that when there is an assessment, that you will have something to study from. So you will have to do a variety of different tasks here, drawings and descriptions, based on what we go over here. We get that out of the way now. And let's go over these objectives. See if we could grab a pen too. Nope, there it is. And now we have a pen and we can write. Perfect. So the objectives. After finishing today's lesson, you will be able to describe the relative size of bacteria and viruses compared to eukaryotic host cells. Eukaryotic cells are the cells that you have. What I mean by that is these are the cells that have nucleuses. The DNA has a membrane around it known as a nucleus, where these other two do not. The DNA, in a sense, is just floating around, well, naked. All right. So we will also name viral structures and describe their functions in viral infections, and we will describe the viral life cycle. So how big do you think a virus is in comparison to bacteria and to red blood cells? To help us, we have this website. This one is garbage. It did not work when I attempted to launch it. So let me show you the one that does work. In perspective, they're showing you the width of a human, not a human, they have a human hair here, but the width of a needle. Let me show you a human hair and look at this arrow as we go move on to smaller and smaller items. The human hair, much, much smaller, and here it is, between here and here, moving left to right. And on, well, living within the hair are dust mites. And here's some ragweed. Ragweed is a type of pollen that might cause hay fever. And let's take a look at these other structures. Let's zoom in. Here is a lymphocyte, a type of white blood cell. Here's your red blood cell. Baker's yeast, used to help make bread and other items. E. coli, that's a bacteria. Look at the scale of the bacteria versus your white blood cell and your red blood cell. Staphylococcus, another type of bacteria, but now look at the viruses. And I bet you didn't even notice these little guys there before. Those are the rhinoviruses for the common cold. Let me actually zoom out and see if you could even see where they were. See, there's the Ebola, and you don't even see those little rhinoviruses. They're tiny. So now let's slide this out of the way and proceed through some more slides of the PowerPoint. Hopefully it works smoothly. It didn't earlier. This is now my third recording of the same material. All right, so here's one eukaryotic cell. Again, it's the type of cell that has within it a nucleus, meaning that it has that double-stranded DNA in there. And that double-stranded DNA is going to send out RNA to ribosomes, which are then going to make proteins. That's going to be important later. Now, here is just one bacterial cell. And these numbers down here are significant because it's showing that from side to side, we could fit 20 bacteria in this cell. So why can't a eukaryotic cell fit 1,000 bacterial cells instead of only 10? Well, we don't just have left to right. We also have up and down to consider. And we also have depth. We have to consider how deep it is. So we can fit a large number of cells, these bacterial cells, inside these eukaryotic cells. All right, now let's change it. And our main focus is one bacterial cell versus viral cells. If you notice, this number here is 20 times the size of this one. So how many viral cells can fit inside? And I think my math on the previous slides, let me go back to those previous slides. I did say 20, 10 times bigger side to side. Now let's go forward to where we were. So from side to side, we should be able to fit in 20 viruses, but also consider top and bottom and the depth. This is a three-dimensional world, after all, isn't it? So why can a bacterial cell fit 8,000 viruses instead of only 20? It's a three-dimensional world. So now, how many of these guys can fit in there? Well, in one eukaryotic cell, we could fit 1,000 bacteria and 8 million viruses. Once again, we're looking at scale here. So if right here we're looking at eukaryotic cells, such as your body cells, prokaryotic cells, varying in different types, are smaller. Viruses, varying in their different types, are also smaller. And proteins by themselves are smaller than that. So what is a virus? A small infectious agent that cannot reproduce on its own. Viruses need a host cell machinery to replicate. So standing alone, virus cannot make more viruses. The only way that they can is if they infect a cell. Kind of like a parasite can get into an organism in order to feed off of it. Well, it's not even actually feeding. It's just hijacking the machinery to make more of itself. So, what might a virus need to replicate? Well, if a virus has DNA or RNA in it, that will come up later. It needs a way of making more of that. It's going to make proteins. And in some cases, it's going to steal some pieces of the cell that it's exiting. So the structure of a virus from inside out. All right, so we got the genome. So that's the genes. So that's the DNA. Sometimes might be the RNA. We have something called the capsid, and that's a protein coat that covers the genome. So it's going to protect it. And the envelope. Sometimes it has an envelope, and that's a lipid protein coat that covers the capsid, and this is a part that is stolen from the cell that it is exiting. So, moving on, viruses with just a genome and a capsid are called naked. So in other words, they do not have this. The envelope is sort of the clothing. Viruses with a genome, a capsid, and an envelope are called enveloped. Here's a picture of them. Here is a cut open virus. It has the envelope, it has a capsid, and it has that genome. It has DNA or RNA in it. And this one is not cut open, and it looks kind of sinister. But all it is that capsid, and inside we do have a genome. So inside the capsid, we can see DNA or RNA, and those are the codes to make proteins to therefore make more viruses. The capsid itself protects the genome. It's composed of protein, and it contains enzymes needed for replication. The envelope protects the capsid when present, so not all have it, composed of protein and lipid. Remember, lipids are fats. Our cell membranes are mostly lipids, and they have proteins in them, and contains proteins needed to attach to host cells. So these proteins, these guys right here, are going to be part of the charade to trick a cell into taking it in. And they do have a life cycle. They need to attach to a cell. They need to enter. They need to hijack the host cell so that it could then be tricked into replicating, and it needs to exit, and therefore then it's going to infect others. So how depends on whether the virus is naked or enveloped. They have different strategies. Come on, you were doing so good. Next slide. There we go. This is how the naked viruses enter the host cell. So the naked virus might inject their genome through the host cell membrane. For example, viruses that infect bacteria, bacteriophages. So we do have some we do have some viruses that will actually attack bacteria. So we're not the only ones under attack. And the naked viruses might be taken up by endocytosis and then punch holes in the endosome membrane. So we're going to show you that. This will become animated in just a moment to show you both. So here's a bacteriophage. There's the poliovirus. So in this case, it's just being injected in. The capsule's left behind. But in this next case, on the bottom, come on, start to move. We're having another delay here. There we go. Instead of being injected, it kind of pushes its way in, and if you notice, it took some of the membrane with it. Now, on the other hand, the enveloped ones, these guys are important. Because the enveloped viruses also has a membrane, it might fuse with the host membrane. Remember, this membrane came from a previous host, so it's going to resemble this, and it might just mesh right in, and they're calling that fusing. And the enveloped virus might be taken up by endocytosis once again. Let's see how that works. So in this case, it just blended in. It was fluid within, and the inside just popped out on the other side. In this case, similar to the last one, it is wrapped around by the cell membrane of the host, and it is brought in, and then it releases its genome. All right, so how naked viruses exit. So now that they went in, they need to replicate. So they took over, they replicated, and now they need to exit. So the host cell fills with mature viruses, and it bursts. And we call that lyse. Lysing means to break. Now, how envelope viruses exit? Well, the new envelope proteins are collected in the host cell membrane. So they're getting stuck there. The new capsid proteins in genome collect below the envelope proteins, and the new envelope virus forms and buds off from the host. So it ends up right here pushing against it. And you can see this is a time lapse. Now it's starting to take some of that cell membrane with it, and now it is completely coated with the cell membrane from the host. And when it goes and it meets another cell that had the same style cell membrane, it's just going to blend in again and do more damage. Now, I found a video that's actually a little bit better than this, so hopefully I can stop this and bring the other one in. And I like this one in comparison. Let's make it larger and play it.
So let's say that this guy has the
Yuck.
flu, could be any flu, and here's a droplet from his sneeze containing, if we move in and take a really close look, you see each one of those little purple things is a virus. And there are a lot of viruses floating through the air, some of which go inevitably up this unfortunate man's nose.
How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay, I am talking with medical illustrator David Belinsky, who designed this video for Xyrus, a research company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah, this is a key, this is a key, this is a key.
Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus. So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface where they get new keys, and then boom! Here they come. This is an eruption of virus after virus after virus after. Only one virus entered the cell, but how many came out? Well, millions. Millions. So if one virus can produce 1 million babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1 million viruses is just a drop in the bucket when you have 100 trillion cells. And anyway, remember, you do have your own immune system, which when it sees a virus usually kills it. So while the virus does multiply fast, with any luck, your immune system will work just a little faster. So, yes, viruses, all viruses want to spread. That's what they do. But most of the time, we do keep them in check. Most of the time.
So what do you think of that? Let me get this guy out of here. Maybe not. Yes. All right, we'll just finish it up right here. So let's recap. All right. Viruses, there are two different types. There's the naked one and there's the one with the envelope. They both ultimately take over a cell and then destroy the cell in the process, only to infect others. So work on that worksheet. Get it into me by the due date that you will see posted with this video, and I will return the favor after the due date and return you a copy that has the answer key, and we will move on from then. Keep washing your hands, everyone. Keep washing your hands. Be safe.